Good afternoon. Just a real quick plug that I and a lot of my cohorts are cool researchers, chemical origins of life. And I'm a proud member of the Center for Chemical Evolution. Kind of like what Bradley was saying, that we understand that biopolymers are the constituents of life. And we look at polynucleic acids, polysaccharides, polypeptides, and really we look at proto-poly-nucleic acids, poly-peptides. And what I'll be talking about are proto-poly-peptides. And we really like to embrace the evolution and the transformation of these systems. So to put the proto in the proto-poly-peptide, we have to motivate that, we have to consider some of the challenges of poly-condensation of amino acids in an early Earth environment. The direct condensation does require high temperatures. It can also be achieved with a complex catalyst that may not be prebiotically plausible, and of course, sometimes called the DKP trap, this cyclic dimer that occurs after you have your first condensation that is a trap towards further linear polymers of amino acids. So we look in two places, meteoritic samples and also spark experiments are two of probably many, and we have to acknowledge that the prebiotic molecular inventory is more diverse than just what we have in our bodies right now. So this an analog to amino acids, hydroxy acids are quite similar, except they have a hydroxy group and therefore can form ester bonds instead of amide bonds, which are chemically distinct. And these are plausible, of course, because they do appear in samples like the mercury meteorite and do appear in abundance in spark discharge model prebiotic systems. And so what we have found in our search to have this proto-poly-peptide is an ester-mediated amide bond formation. So in evaporative conditions at mild temperatures, 85, and we've explored some other temperatures as well. But we, as I've mentioned here, the direct peptide formation can lead to the cyclic dimerization, kind of the end of that cycle. We don't see, in nascent conditions, we don't see polyglycine or polyamino acids. But hydroxy acids, especially lactic acid, what I'll be talking about a lot today, they readily form polyesters in these conditions. So if we combine these two, this copolymerizing system, at this ester, the carbon of this ester site is activated for nucleophilic attack by the amino acid, and we get this what we call exchange. And this can be repeated, so this esterification exchange, until we get something that has a mixed amide and ester backbone that we refer to as a Depsipeptide. And this can go on indefinitely, and we get shown here a wide variety of sequences and compositions of oligomers with appreciable length. And this can just go off like a shot. So we get these copolymers that, like I said, have ester and amide backbones. So way back to the Bernal's hypothesis, but just that minerals have been this ever-present theme in prebiotic chemistry. Their ubiquity, their diversity, and the unique dynamics that go on at the interface necessitate their consideration. And especially in evaporative systems, where we drive off water to promote condensation, in dilute systems, which are very plausible, that the substrates will almost like always be in contact with the molecules, and their interactions need to be considered. So focusing on the composition of Depsipeptides, you can have, for say a 6-mer, you can have anything from a holy peptide to something a polyester. And of course, the Depsipeptides are somewhere in between. So using mass spectrometry, we can have a reaction and then understand the relative abundances of the different compositions. Here, the dominant species present in this population is a 6-mer. 6-mers has 3 lactic acids, and in this case 3 glycines. And what we can also do is compare different conditions. So the addition of, in this case, fumed silica presents a completely different composition distribution. Here we see that the dominant species only contains one hydroxy acid or lactic acid. So this is much more peptide-like in character. And through other mass spectrometry means, it's likely that this one hydroxy acid is actually on the end. So we have a core of amino acids, a peptide. So this enrichment of amino acids is very interesting and occurs in the presence of silica. So just some quick experimental notes. This reaction, a lot of them have been doing it at 85 degrees Celsius, an open vial, and this is in acidic pHs. And this not only works for this enrichment of amino acids, not only occurs for a glycine lactic acid system, but also alanine, so a slightly more complex amino acid. At 85, we see that from the black herd and the black and red inset, that a lower atomic mass is actually something that is more peptide rich, because nitrogen has a lower atomic mass than oxygen. And for alanine, again, even at higher temperatures, that the distributions are distinct. And there are other ways to verify this amino acid enrichment in the presence of silica. We can look at NMR, and I won't go into how we assign these peaks, but we can identify these key bonding motifs, some associated to esters, we see in the first one, that is present in the no substrate control and is absent in the silica samples. Something that corresponds to internal amide linkages is much more abundant in the silica-containing samples than in the control. And another example of a motif associated with polyesterification that is present in the control and not in the silica-containing samples. So that was very interesting. And so our next step, and to kind of understand maybe how this was happening, was to kind of break up the reaction into steps. So first we were reacting the silica with the hydroxy acid lactic acid, incubated it, then washed it to remove maybe any non-covalently bound species, and then added our amino acid afterwards. And what we got was a population of Depsipeptides that had no esters, so they only one lactic acid on the C terminus. And so that if we were to form these these amides, we would have had to have had an ester present to be the activator. And so that kind of got us thinking of what may be occurring on a hydroxyl terminated silica surface, that we could perhaps have formed these interfacial esters. That when the glycine is present later, these were active sites, as I expressed earlier, for ester-amide exchange, and that we were able to form a depsipeptide. So to begin to confirm this, we thought about modifying our hydroxy acid. So now it's a methoxypropiononic acid. So this is now a monofunctional molecule that cannot participate in esterification, though for other purposes, such as the acidity and the pK of the acid are quite similar to the lactic acid. We reacted that with an amino acid, and as it turns out, in the absence of silica, we do not get only trace amounts of the condensation products. But in the presence of silica, we get up to 4 or so glycines attached to this 1,2-methoxypropiononic acid. And this suggests that the silica can participate in the formation of an ester, therefore an active site that can allow for amide synthesis at the temperatures. And also, we can look at other substrates, some minerals, TiO2 and hydroxide, and see that titania does produce some condensation products, and aluminum hydroxide does not. And I think that's interesting. So just to conclude, that we are able to form depsipeptides in the presence of silica, and that the presence of silica enriches the population in amino acids, both glycine and alanine. We can be sure about that because ester linkages are reduced in the presence of silica, and amine linkages are increased. And that it is suggested that the amide synthesis in the presence of silica is actually facilitated by a interfacial ester. And so going back to the last slide where we showed some other minerals, what characteristic of the mineral would be most predictive of its ability to enrich the system, a peptide-forming system, Depsipeptide-forming system with amino acids, is unclear right now, but that's the target of present work. And that's all I got right now. Thank you.
Questions? Okay, cool. That was a great talk. Thank you. I had a question about, a kind of comment question about silica. That's because we think that silica availability on Earth has changed a lot through time. Now it's controlled a lot by biology because there's shell formation. So do you think that because silica might have been more available back in Earth history, would that affect your results?
So I've actually, a lot of these studies have done with fused silicas, which I wouldn't even consider formally a mineral. That's, pyrolysis created. The question of the abundance of silica, I'm not exactly sure. I think there are probably other minerals that are far more abundant. TiO2 may have been one. I don't mean to say that silica is the only way that we can enrich. I mean, you don't have to use any mineral to enrich Depsipeptide systems with amino acids. You can cycle these. You can add more of one monomer. But the chemistry with a mineral and a Depsipeptide system needs to be studied and is distinct from a mineral and a purely amino acid system, which has been looked at extensively in the past. And so I think the fundamental mechanisms are distinct.
Yeah, time for one more question. Oh.
Yeah, so what is it that's ultimately limiting the length of the oligomers that you can form?
One of them could be, besides the monomer, I think solubility of the. If we get too large, then the oligomers may conform in some way where they're not able to, or the ends are not accessible for reesterification. Another component is that the lactic acid or the hydroxy acid is volatile to some extent, so that could be leaving the system. Also, the lactic acid will polymerize with itself, so the availability of that may also be limited, a limiting factor.
Okay, so let's thank Aaron.
Thank you.